Okay guys, in this video we're going to continue our discussions on thermochemistry, and we'll also discuss a bit later calorimetry, which refers to experimental techniques for measuring the heat flow during a chemical reaction. Well, in this first part we're going to talk about the temperature dependence of chemical reactions, and so the main point is that if you carry out a reaction under conditions of different temperature, you're going to find that the enthalpy change is not the same at the two different temperatures. And so we'll go back to this idea that when you, in order to change the temperature of a substance, you need to add heat. And so what this formula here shows is it's the enthalpy at some temperature T2 is the enthalpy at some temperature T1 plus a correction factor involving the heat capacity. And so these are the problems that we looked at before, where we were, say, heating a gas up between T1 and T2. Under conditions of constant pressure, we need to know the constant pressure heat capacity. If it's a temperature-dependent function, then we need to integrate over that heat capacity. So this would be for a pure substance, just simply heating it up. We can take this formula and modify it for chemical reactions. That is, the change in enthalpy for a chemical reaction at a temperature T2 is equal to the change in the enthalpy at some temperature T1 for that reaction, plus a correction factor involving the change in the heat capacity of the system as it undergoes that chemical reaction. So this delta Cp is the sum over the heat capacities of all of the reactants and products multiplied by their signed stoichiometric coefficients. So this number, nu j, is negative for reactants and positive for products. Here's an illustration showing you what this formula does. So we have some chemical reaction for which the enthalpy of reaction is known at a temperature T1, but we'd like to know what is the enthalpy of reaction at a different temperature T2. The problem is our thermodynamic tables are not tabulated for T2, but they are tabulated for T1. That's why we know the delta H at T1. So think of this point right here as your initial point, and this is the desired final point. Well, we can't calculate the change directly, so we need to use essentially Hess's law, or what we're calling now Kirchhoff's law. So what we're going to do is we're going to take our reactants and we are going to cool them down to T1. Then we're going to undergo the reaction to make all of our products, and then we'll heat our products back up to T2. Hess's law tells us that this leg of the cycle is going to be equal to the enthalpy changes for the sum of the other three legs. And so you can go either way. Because enthalpy is a state function, it doesn't matter if you go directly from your initial to final state or if you take this other path. And so that's what's encapsulated here in this formula. This is the reaction. This is that vertical arrow that we knew. This is the vertical arrow that we wanted. And then this is the cooling and heating. Okay, so the minus sign in this formula corresponds to the cooling of the reactants, and then the positive portion of the change corresponds to heating up the products. And so here's an example problem. Let's see, what are we being asked to do? We're given the standard enthalpy of formation at 298 for water vapor, and we're being asked to estimate this value at 100 degrees Celsius, and we are given the following heat capacities. So what Kirchhoff's law tells us is that the enthalpy of reaction, here I'm going to go ahead and write the reaction first. So we've got H2 gas plus 1/2 O2 gas gives us H2O gas. So that's the reaction that we're dealing with. we want to know what is this reaction enthalpy at, let's see, 100 degrees Celsius would be 373 Kelvin. That's going to be equal to the enthalpy of reaction at 298 Kelvin. And so we know this value. And then there's going to be a correction factor involving an integral from 298 to 300 Kelvin, which involves the change in the heat capacity. So the change in the heat capacity in this case is going to be the heat capacity of the water vapor minus the heat capacity of the hydrogen gas minus 1/2 the heat capacity of the O2 gas. And I should have made all of these molar quantities. Like that. In this case, the heat capacities are all assumed to be constants over this temperature range. That's not necessarily true. In fact, they could be functions, in which case we would be calculating a function of temperature here that we would then integrate. In this case, it's simpler. We're just assuming that everything's a constant. So we can throw in these numbers here. 33.58 minus the number for water, 28.84 minus 1/2 the number for oxygen, so we'll calculate that real quick. I'm getting minus 9.9 for joules per mole Kelvin. Okay. And so since that's a constant, it's going to come outside of the integral, and this formula is going to simplify to that. Right, that's because this is a constant. It comes out of the integral. So we can plug in the numbers here. Minus 241.82 kilojoules per mole. We'll have minus this negative 9.94 times 10 to the minus 3 kilojoules per mole Kelvin. And then the temperature difference is 373 minus 298 Kelvin. So we can calculate that. And it's really not that large of a difference. Minus 241, assuming I didn't make an error here, minus 241.07 kilojoules per mole. Right, this is a pretty small number that I got. So assuming that I did the calculation correctly, we see that this difference in temperature here is not very large. Right, the number that you had before is fairly similar to the number that we have now. In any case, that's how you perform these calculations. Let's see what we have next. Okay, so it looks like we're going to move on to to calorimetry. So calorimetry is a set of techniques. There's a whole bunch of different calorimetry techniques for performing the measurement of heat associated with a different physical or chemical process. I've only got 4 of them listed here. The first one, bomb calorimetry, occurs at constant volume, and so we use a composite system, and I'll show you a figure in just a second, where the chemical reaction itself, the bomb vessel as it's called, and then the water bath that surrounds the bomb vessel are all considered to be part of the same thermodynamic system, and then the surroundings would be everything else. Here's a schematic diagram. We have this gray box here. This would be so this is a cutaway version of it. This steel metal container here, this is the bomb device. Okay, so it's a constant volume. It's very rigid material. Inside there's a sample holder and 2 electric leads. And so what you do here is you take your sample and then you pump it full of oxygen. Okay, so you have the sample in the presence of an excess amount of oxygen at high pressure, and then you've got these 2 electric leads that allow you to ignite the sample. So that's why it's called a bomb, because you're basically burning up everything inside. Typically that's going to be an exothermic process, and it's going to heat up the water bath surrounding the bomb device, and the temperature of that water bath will be recorded. The temperature increase will be reported. Now there's a trick to this in the sense that bomb calorimetry is technically considered an adiabatic process. Recall that adiabatic means that no heat is transferred. And you might be scratching your head here, well, if you're burning something up and the temperature is changing, then how can it be adiabatic? Well, the trick with that has to do with how the system is defined. Like I said before, we define a composite system consisting of the reaction, the bomb vessel, and the water bath that surrounds it. So our system is the water and everything that's within it. The way that you make this bomb calorimetry experiment adiabatic is you use a computer to control the temperature of this outer water jacket. Okay, so as the temperature inside goes up, you simultaneously raise the temperature of this outer jacket. And if the temperature inside is the same as the temperature outside, then no heat will flow from inside to outside. So that keeps all of the heat of the reaction, it keeps it all contained within. So as a consequence of that, because it's isochoric, work is 0. Because it's adiabatic, heat is 0. So the internal energy change of the composite system is also 0. Now this doesn't mean that energy is not moving around within the system, it's just that the energy change of the composite system is controlled to be 0. Now you've got 2 subparts of the system. You've got the chemical reaction itself, okay, which is what we're essentially trying to find, is the energy change of the reaction. And then you've got the heating associated with the calorimeter itself, right? So the bomb device and the water jacket, its temperature is increasing. Those parts of the system have a heat capacity, and so this portion is associated with heating up the bomb calorimeter and the water. This portion is the heat associated with the chemical reaction itself, and these 2 things must add up to 0 for this constant volume adiabatic process. So here's how bomb calorimetry works. You need to know the heat capacity of the calorimeter itself. So what you typically do is take a standard, usually benzoic acid, for which delta U is known. Okay, so you take a known amount of benzoic acid, burn it up, measure the temperature change, use the known value of the delta U for benzoic acid to then find the heat capacity of the calorimeter. Okay? What you're doing is you're calibrating the calorimeter. So once you've found the heat capacity of the calorimeter, then you can now take that value and then use an unknown sample, whatever you're interested in studying. It could be food, it could be a drug, it could be a fuel, it could be a cosmetic, any substance that you want to know the delta U for. You would then burn it up inside the bomb calorimeter, measure the delta T. Using the calibrated value, you can then find the delta U for the unknown sample. In this problem, they're basically asking us to do a calibration of a bomb calorimeter. So calculate the heat supplied to a bomb calorimeter with no sample by running a current of 10 amps from a 12-volt battery for 300 seconds. If the temperature rise of the calorimeter in the water bath is 5.5 Kelvin, calculate the calorimetry constant. So in this case, we don't have a sample. We just simply have that the delta U for this process is the heat capacity of the calorimeter times the delta T, which equals 0. Well, here, this is equal to the heat supplied, I should say. So what we've got here, you've got 10 amps, so 10 coulombs per second. You're running that for 300 seconds, and then it's 12 volts, so 12 joules per coulomb. So this is the heat that's being supplied to the calorimeter. So that's 3,600 joules. And then to calculate the heat capacity, so this is equal to the heat. There's no work done in this case. So the heat capacity will be the heat divided by the delta T. And I'm getting 6,545 joules per coulomb, joules per Kelvin, for the heat capacity for the calorimeter. Okay, so you could consider this to be the calibration run, where you determine the heat capacity of the calorimeter. You could then take this value and then use it to find the using this equation, that delta U equals 0 equals CV delta T plus the enthalpy, the energy of reaction for some unknown reactant. Now that you know that value, you can then take an unknown sample, measure the delta T for the combustion run, and then find the energy of reaction. In the laboratory portion of the course, in the corresponding laboratory to this lecture course, you'll employ bomb calorimetry to measure the internal energy change associated with burning Smarties candy, and you can use that then to compare to the nutritional effects. So further discussion on this will be held in the laboratory whenever you take that. We're going to move on here to the next type of calorimetry, constant pressure calorimetry, which is also often called solution calorimetry. In this type of calorimetry, instead of using a sealed device to burn up a sample, the chemical reaction that you're studying is open to the atmosphere. And so this is useful for studying phase transitions and dissolution reactions, dilutions, acid-base chemistry, biochemical reactions, anything that's open to the atmosphere. Here is the procedure is the same. That is, you consider the calorimeter, its water bath, and the chemical reaction to be part of the same composite system. You have an adiabatic process by monitoring the temperature changes, And so the enthalpy change for your composite system is 0, and your composite system consists of two parts. One is heating up the water bath and the calorimeter itself, and the other part is the chemical reaction. And so again, you need to calibrate your calorimeter to find its heat capacity, and then you can use that value and measure the delta T for an unknown reaction. Here is a picture showing a constant flame calorimeter, which is kind of neat. So you can actually study different fuels using a calorimeter like this that's open to the atmosphere, where this whole apparatus is immersed inside of a water bath. You send a known amount of fuel to be burned inside the chamber, and then you measure the temperature change of the water bath. And you use that data to determine the enthalpy of the reaction. Another type of constant pressure calorimetry that you've probably seen before in your general chemistry laboratory experiment, so-called coffee cup calorimetry, which is frequently used in undergraduate labs to measure the enthalpy change of various chemical reactions. Typically an acid-base neutralization reaction is studied. Another type of calorimetry that's very precise and used frequently in materials chemistry as well as biochemistry is called differential scanning calorimetry. A differential scanning calorimetry uses a setup that looks like this, where you have a sample cell and a reference cell, and they are controlled by the temperature of which are controlled by two independent heaters. And so you may have a material in here or a chemical reaction that's going to take place, and what you do is you scan the you linearly increase the temperature of the sample and the reference by supplying heat. And because there's something inside this sample that's not inside the reference, it usually will require more or less heat to keep the sample at the same temperature as the reference. And so that's the idea of differential. It means you're comparing two things. And then the fact that it's a scanning calorimetry, that refers to the fact that you're scanning the temperature, you're increasing the temperature, and then you're monitoring how much extra heat does it require for me to, or how much less heat does it require for me to keep the sample at the same temperature as the reference cell. And so here, this is depicting that the temperature is usually linearly scanned. So you have some starting temperature. It increases as a function of temperature, and then alpha is the scanning rate. So this is the temperature change that's going to occur. The sample cell is going to have a higher heat capacity than the reference cell, and that usually requires extra heat to be supplied, extra power from the heater to maintain that temperature. And so that excess heat that you need to supply to keep the two cells at the same temperature is expressed here. The excess power for your sample cell times the time that it takes, or times the time that you're at. Okay, and so you can calculate this excess heat capacity by taking the excess heat and dividing by the temperature change. You do this as a function of time, and so if you plug in for the excess heat, this power times the time, you'll find that the time cancels, and so you can just monitor the excess power divided by the scanning rate. And then this is what you monitor. This is what you measure as a function of time. You measure the excess power, and then once you know that, you can then integrate this excess power with respect to temperature to calculate the enthalpy change for whatever process is occurring. It could be heating, it could be a reaction involving a protein denaturing. That's what's being depicted here in this curve. They're studying a protein at a particular pH. It maintains its native structure up until about 45 degrees Celsius, whereupon it starts to denature. It undergoes an endothermic conformation change, and so you can monitor the heat capacity as a function of temperature, and then you can integrate this to determine the enthalpy of the denaturization reaction. So you can use DSC to study various material properties, including properties for biomolecular systems. And the last type of calorimetry I wanted to talk about, this is the isothermal titration calorimetry. This is kind of a neat method. It's also a differential technique, but you can think of it as a souped-up version of the isobaric calorimetry. That's going to be more precise. And so, again, it's a differential calorimetry technique in the sense that you have a reference cell and a sample cell. In the isothermal calorimetry, what you do is you dose your sample at regular time intervals. Okay, and so this causes, like, each time you add some reactant, some more reaction occurs. And so what you wind up with is monitoring the power required to maintain the temperature between the reference cell and the reaction mixture cell. And so I kind of think of this as a combination of DSC and isobaric calorimetry. Here's an example of what kind of data you get out of it. You see these peaks in power. So this is each time you add a dose of the reactant, and then in order to calculate the delta H, what you need to do is add up you need to add up the area under each of these little power spikes, and that's what gives you the enthalpy change for your reaction. Well, I'm going to go ahead and stop this video here. We'll pick up in the next one with a bit more abstract discussion of thermodynamics. That's going to involve quite a bit of calculus.